Hello again, students. You had lots of fun in the last chapter, chapter 2. Well, in the last chapter, you are learning all the organelles and all structures that were related to the cell, both animal and plant cell. Now, one of the outstanding features of a cell was the plasma membrane, wasn't it? Now, today's chapter, we will be delving deeper into the plasma membrane. Well, when you were learning before this, you realized that the plasma membrane was controlling all substances which move in and out of the cell. But you never knew what caused these substances to move in and out, and how does the plasma membrane control? Well, some of you might have questions. Why is it that a water molecule can enter directly into a cell, allowing it to explode? But why doesn't the same thing occur to maybe starch molecules, protein molecules which are larger? So all those questions that you have had in your mind before this in the previous chapter would be answered today. Now let's get into our lesson for today, which would be movement of substances across the plasma membrane. Dear students, most of you would have remembered the diagram of a plasma membrane as a single line on a microscope slide. But now, there were two gentlemen named S. Singer and G. Nicholson. These two gentlemen back in the 70s, they gave a model for the plasma membrane. They said the plasma membrane would look like this. And the model that they came up with was called as the fluid mosaic model. Fluid mosaic model. Wonder, why did they call it fluid? Fluid is a term that is used for anything that can flow along. Now, a mosaic model. What is the meaning of the word mosaic? If most of you are familiar with Islamic architecture, if you just go across the road, you can see on a mosque, there will be a mosaic architecture, meaning which you can see that on a single plane, there are two colors, one a brighter color and a darker color. And from a far view, we would see that they are protruding between each other. Now that is a mosaic model. Now this was the model that was termed by Singer and Nicholson. Now we're going to take a look into the exact structure of a 3D plasma membrane. As you can see, the plasma membrane consists of a phospholipid bilayer, glycoproteins, proteins, carbohydrate chains, cholesterol molecules, and a lots of carrier and pore proteins. Now the three-dimensional structure might look quite intrinsic for you, but don't worry, I have a simpler version later. As we have absorbed the molecules, let's take a look at the fluid mosaic model that was given to us by Singer and Nicholson. Dear students, check out the word fluid. The word fluid refers to a structure which is not rigid or static, but form a dynamic and flexible structure. The word mosaic, however, describes the protein molecules that are found embedded wholly or attached partially to the surface of the plasma membrane. These proteins float about freely on the bilayer to form an ever-changing pattern found on our detailed plasma membrane. Therefore, we can conclude that the plasma membrane has a fluid characteristic which allows the cell to be flexible. As you have seen earlier, fluid mosaic, two words that are very detailed. Now, it is very important for you to understand and really explain well between these two words. Now, let's imagine. I'll ask a question to you. Now, does a cell look like a watermelon, or does it look like a 50-cent coin? A watermelon or a 50-cent coin? How does it look like? Some of you might have said the 50-cent coin. Why? Because most often you see a plasma membrane surrounding a cell drawn as a two-dimensional structure. Well, that's a drawing that you draw. In three-dimensional reality, the cell, however, looks some sort like a watermelon. Now imagine that we have the shell. This is the plasma membrane. The shell of the watermelon is the plasma membrane. And if I cut the plasma membrane, if I cut the watermelon into two, I can see that on the inside of the watermelon, it is filled with a red juicy fruit. Now if you scoop all those things away, what will be left would be only the skin, right? That's how our plasma membrane is. And on the inside, it will be filled with cytoplasm. And the seeds which float around, those are all the organelles which are suspended inside the cell's cytoplasm. Well, today, we are only interested in looking at the plasma membrane itself. So let's see. If the plasma membrane was rigid, what is the meaning of rigid? Now, take for example, this pencil that you have in front of you. Can I change the shape of this pencil? I can't. I cannot change the shape of the pencil. No matter how I try, it is not flexible. It isn't flexible. Therefore, I use a word rigid. It is a very rigid structure. Now, if I wanted to change the shape of this pencil, what would eventually occur is I would snap it into two, right? Therefore, this is the meaning of the word fluid. A fluid structure is a structure that can move freely about, not being rigid, can move freely. But then again, Mr. Singer and Nicholson had a big worry. They wanted to know if the cell membrane could move so fluidly, wouldn't it be too fluid that it cannot support the mass of the cell and eventually it will burst? Therefore, they said, something in the plasma membrane must be making it stable. So what was it? And they found out that the protein structures which are attached to it, they make stability for the plasma membrane. Therefore, the plasma membrane is fluid, but it is not too fluid because it will be controlled by the proteins and other structures embedded. Now, let's take a look at a simple diagram of the plasma membrane. Most of you will be asking a question, come on sir, if it is a three-dimensional structure of a plasma membrane, how am I going to draw it? If they ask me in exams, don't worry about it. Why? Because I've got a simpler version for you. The one that we all can draw, and if you label it accordingly, then it would be accepted in your examinations. Well, let's take a look at this simple structure. Now, as you would have seen earlier in the three-dimensional structures, the plasma membrane consists of a phospholipid bilayer, which means two layers of phospholipids. Now, the two layers of phospholipids will be embedded, or things will be attached into them. What are those things that are attached or embedded to them? They are the proteins. Now you can see that the proteins that are attached are not only for a single purpose, but they have different functions. For example, we have one here, we have another here, and we have another here. These three proteins are of totally different importance. Now let's label each one of them. Now, as you already knew, this is known as a what? Proteins. Very good. Now, these proteins have certain specific function as of to stabilize the plasma membrane. Remember we were speaking earlier about Nicholson and Singer's theory? Yes, the plasma membrane is fluid, but it needs something to stabilize the fluidity of the plasma membrane. Well, you have your proteins. Some proteins, however, are specially structured. That means they have a special three-dimensional shape and structure which allow little molecules to come and attach into them. Now take for example, let's say we have a glucose molecule. These glucose molecules can come and attach to these carrier proteins, which will then open up and allow the glucose molecule to pass through the plasma membrane. So once again, students, if I was to label this structure, what would it be called? Yes, you are right. It is a carrier protein. Let's go to our next structure. Now, this structure has a very thin pore in between it. A pore is a smaller version of a hole. Now, this little pore allows ions to move in and out of them, and therefore we call it as the pore protein. Now students, I came across a few students who came up with a label, and during their exam tension and all, what they did was they started to label this as a protein pore. They labeled it as a protein pore. Now what's the big difference? Come on, protein pore, pore protein, both the words are there, it's got to be right. But then again, it's wrong. When you say it's a protein pore, it means that proteins will travel through this pore. But if you say it is a pore protein, then it means that the pore is made up of protein. So there's a big difference between them. And remember never to label it wrongly. As you can see the components of the plasma membrane in the image shown, you will see that the phospholipid bilayer separates both sides of the membrane, namely the inside and the outside of the cell. And you can find cholesterol attached to the plasma membrane. This cholesterol links the fatty acids which are found in the phospholipid layer. And eventually, it stabilizes and strengthens the plasma membrane itself. Without these attached cholesterols, the plasma membrane would never hold the mass of its cells. And the cholesterols make the plasma membranes more flexible and less permeable to other water-soluble substances, therefore creating a semi-permeable characteristic of the plasma membrane. Well, you can also see there are other structures, for example, glycoproteins which are attached to the plasma membrane. The glycoproteins function as a part of identity to the cell. Therefore, the cell would recognize each other based on proteins or glycoproteins which are attached on the plasma membrane. As you can see in the image, the plasma membrane has a phospholipid bilayer which separates both sides of the membrane, thus allowing the membrane to differentiate between the outside and the inside of the cell. As you can also see, you will find that there are cholesterol molecules attached to them. These cholesterol molecules link the fatty acids which are found in the phospholipid membrane. This allows the cholesterols to stabilize and strengthen the plasma membrane. Therefore, the plasma membrane would become more stable and more flexible thanks to the cholesterol molecules. These cholesterol molecules also will prevent other water-soluble substances from allowing entry into the cell. Therefore, these will also create eventually a special characteristic of the plasma membrane known as semi-permeability or selective permeability. And as you can also see, there are proteins, and certain proteins have different structures. For example, as you can see on your slides, the pore proteins and carrier proteins. These proteins are used for transport of small and polar substances such as ions, and also large and non-polar substances such as amino acids and glucose. However, we can see that there are two of them. One is a carrier protein which has a larger shape and structure. Therefore, the large and non-polar molecules will gain entry through the carrier proteins. The pore protein on the other end has a very small channel leading to and out of the cell. Therefore, small ions or small polar molecules will gain entry into the cell and out of the cell through the pore protein. Well, what is this word called polar? Never heard of it before. Well, in other subjects, you would term ions as charged molecules, either negative or positively charged molecules. But in biology, we are more accustomed to use the term polarity and polar, which eventually means the same thing. Next, we will want to discuss the special characteristics found in the plasma membrane. Well, the plasma membrane shows few things. Let's take a look. Plasma membrane shows selective permeability, which means some substances can move across it, but other substances cannot gain entry into the cell. What causes this to occur? Now, the factors that affect permeability include the size of molecules and the polarity of the molecules. Now, remember what we were saying just moments before. If we have a large molecule, it will have to gain entry into the cell following the carrier protein. But if we have a very small molecule, it will gain entry into the plasma membrane through the pore protein. Now, carrier proteins and pore proteins allow different types of molecules based on their polarity and based on their size to gain entry into the cell. As you have already seen the plasma membrane structure, This is the simplified structure, and we have proteins attached to the plasma membrane. Proteins and cholesterols attached to the plasma membrane allow the plasma membrane to gain stability and strength. Well, these proteins not only create stability for the cell, these proteins also create an identity for the cell. These proteins will also be known to you later when you enter into Form 5 as antigens. Now let's say, for an example, a bacteria decides to attack us. The moment the bacteria is into our body's white blood cells will determine that it is a pathogen, and our white blood cells will identify that these do not belong to us based on the proteins found on its plasma membrane. That means every plasma membrane has its own identity thanks to the protein. And because of these proteins, our white blood cells does not attack our own cells. Next, what we're going to see is, we want to see a conclusion or a small summary about what substances can move in and out of this plasma membrane. Now, focus on the phospholipid bilayer. Now, the phospholipid bilayer consists of the head and the tail. And we can see that between these two phospholipid molecules, there ought to be a little gap. But this gap is very small, therefore allowing only very, very small molecules to squeeze through them. Another point for you. The phospholipid bilayer consists mostly of this region known as the lipids. Lipids are fats. Therefore, whoever can penetrate through this plasma membrane should be of the same class as the lipids. So, whatever small and lipid in nature, will gain entry directly via the plasma membrane. Remember, direct entry, small and lipid-soluble molecules. However, because the gap is so narrow, a special molecule can also gain entry. This molecule is known to you as H2O. Yeah, you're right, it's water molecules. Therefore, water molecules, small lipid molecules, are the VIPs of the cell. Therefore, they can gain entry in and out of the cell without any form of obstruction. Next, large glucose molecules and large amino acid molecules. These glucose and amino acid molecules can only gain entry into the plasma membrane through the carrier protein. Next, the pore protein. Pore protein has a very narrow channel, which allows the movements of small and polar molecules only. Now guess for me, a small and polar molecule. Almost. Ions, boys and girls, ions. Ions are small, and because they carry charge, we term them as polar. So what are the ions that can travel through our plasma membrane? It can be any ion, but in biology, we've got three that we regularly use. Those three ions are known to you as K positive, Na positive, and Cl negative. Potassium, sodium, and chloride ions. So as we have seen the plasma membrane before this, the major characteristic of the plasma membrane, or the main component of the plasma membrane, is the phospholipid bilayer. Yeah, bilayer. Why do we need to have two layers of it? Why not just have one layer? Wouldn't it do the job? There must be a reason to this, and we will have to investigate on it. Now, the phospholipid bilayer holds the key to a lot of things on how the plasma membrane works. Therefore, let's take a look at the phospholipid molecule itself, because each one of these molecules has got some speciality in each one of them. Let's take a look at the phospholipid molecule. The molecule consists of two parts, a head and a tail. The phospholipid molecule head consists of a phosphate ion. Yes, therefore it is polar, and because it is polar, it can dissolve in water which gives it a characteristic of being hydrophilic. The term hydrophilic refers to ability to dissolve in water. However, in the same phospholipid molecule, the tail consists of lipid, which is fats, and the tails cannot dissolve in water, as yes, you have guessed it, because it is non-polar. Now, because it can't dissolve in water, therefore it creates a characteristic stated as hydrophobic characteristic. Hydrophobic refers to the term not being able to dissolve in water. Now, as you can see from the phospholipid molecule, the head and the tail consist of totally two opposites. Whereas the head can dissolve into water willingly, the tail has a fear of water. So the term fear, we call it as what? Phobia. I'm having a phobia towards my biology teacher. The same way, the tails are having a phobia towards water, which means that they will not be dissolving in them. I have drawn for you a simplified version of the phospholipid molecule. Now, it's very important for your exams because the characteristics determine a lot of characteristics of the plasma membrane itself. So, this was the head, and the head was consisting of a phosphate ion. I've given you the formula here. Yes, it is 3 negative because, as you have learned in chemistry already, phosphoric acid would be H3PO4, which gives phosphate ion a negative charge. It is hydrophilic. Now, hydrophilic means what? Ability to dissolve in water. Very good, very good. Because it is polar. Because it is polar. Next, the lipid tails in the phospholipid molecule consist of fats, lipids, which gives it a hydrophobic nature. Why is it hydrophobic? Because lipids can't dissolve in water. Why can't lipids dissolve in water? Because lipids are non-polar. So if you see, students, there are lots of different types of questions that we can ask you. I can ask you, what are the characteristics of the phospholipid molecule? You will have to answer, the phospholipid molecule shows both hydrophobic and hydrophilic characteristic. Now, from that one question, I can develop and ask you more. Why does it show both hydrophobic and hydrophilic characteristic? Answer. The tail comprises of lipid which is non-polar, which can't dissolve in water, therefore it is hydrophobic. The head, which consists of phosphate ions, are polar, therefore they can dissolve in water, therefore they are hydrophilic. So you see, between these terms itself, we have covered a wide range of objective questions and subjective questions. What about an essay question? Now, a lot of students will say, yeah, obviously, you teach me today, I listen to you, and I know what goes on. But come exam, I can't do it. I mean, there's simply too much to explain about in an essay question. Let's take a look. Let's say I have a question which states that, describe the characteristics of the phospholipid molecule. Very simple. Describe the characteristics of a phospholipid molecule. I don't remember everything that I've learned in my text. What I do remember is a structure and 4 key points. Now, based on the structure and 4 key points, what I know, I can offhand get my essay. Watch and observe. A phospholipid molecule consists of 2 parts, a head and a tail. The head of the phospholipid molecule consists of phosphate ions. Phosphate ions are polar, therefore they can dissolve in water, which shows a characteristic of hydrophilic in nature. The tail, however, which consists of lipid, shows hydrophobic characteristics because it can't dissolve in water as the lipid is not polar. Full stop. Now that would easily earn you the 4 marks or 3 marks that would be required from you. Now, if you can go from small notes, from short, small points, to developing in your own words, it's more than good enough. So don't worry about how it would turn out to be at the end, because all you need is to be confident with the keywords, and eventually you'll be able to do it yourself. Next, let's imagine that this is a barrier, and on the left hand side, this is the inside of a cell. Let's imagine this is the inside of a cell, which means these dots are all water molecules. Sir, from where do the water molecules come into the cell? Well, inside the cell, we have cytoplasm, don't we? Well, the cytoplasm comprises of mainly water. On the outside of the cell, however, I say to you again, there is water molecules. Come on, sir. How come on the left and on the right, both sides, in and out, go water? Cytoplasm consists of water. Interstitial fluid consists of water also. Now, imagine yourself as a phospholipid molecule. Now, you are a phospholipid molecule, right? Let's say I let you to sit in the center. And as you're sitting on the center, your tails, or let's imagine our legs, our legs are hydrophobic. The word phobic determines phobia, scared of water. Can you put your legs on the inside? No. So what do? Can we put the legs on the outside? No. Wow, better still. Can we put the legs out like this straight away, just pointing out? Also you can't, because it's three-dimensional. Now that means the tails of the phospholipid cannot face on the inside, and neither can they face on the outside. What do? So imagine this is the head of the phospholipid molecule. This is the tail of the phospholipid molecule. Now I take two phospholipid molecules and bring them here. And as they enter in here, what they do is they clearly push the water away, and so that the tails face on the inside. Now, if that was the case, let's think back and let's imagine that we have the center once again. We have a head, we have another head here, and both tails face on the inside. Now what will happen is the liquid or the fluid on the inside and outside is being separated by the phospholipid molecule. The heads are in the water. Are they happy? Yes, they are, because they are hydrophilic. The tail is away from the water. Are they happy? Yes, because they are hydrophobic in nature. Well, that's a simple explanation on the phospholipid molecule. As we have already come to the end of our first part of this lesson, let's test ourselves to see whether we caught all of it and we have all the information necessary. Let's go to a self-assessment. Question 1. The current structure of the plasma membrane is best described by using the A. Phospholipid bilayer model, B. Fluid mosaic model, C. S. Singer and G. Nicholson model, D. Plasma membrane of plant cell. The answer? Yes, B. Fluid mosaic model. Now the fluid mosaic model was given to us by Singer and Nicholson, but they did not give their name to the model. Question 2. The main component that makes up the plasma membrane of a cell are A. Proteins and carbohydrates, B. Proteins and phospholipids, C. Phospholipids and carbohydrates, D. Proteins, phospholipids and carbohydrates. The answer? B. Proteins and phospholipids. As you have seen before, the phospholipid bilayer and protein molecules which are attached onto the phospholipid bilayer. Let's go to question 3. Which substance can move freely across the phospholipids of the plasma membrane? Answer A, glucose, B, glycerol, C, amino acids, D, ions. Answer B, glycerol. Now glycerol and fatty acids combine to make fats. Therefore, glycerol is involved in lipids. Question 4. When only specific substances can move across the plasma membrane, it is said to be A, semi-permeable, B, impermeable, C, porous, D, selective diffusion. Answer A, semi-permeable. Permeable refers to allowing inside. So permeable means allowing inside. Semi-permeable means allowing inside but not all molecules. Certain molecules will be allowed, certain molecules will not be allowed. Question 5. Why is the model used to describe the plasma membrane called a fluid mosaic model? The protein molecules and the phospholipids float laterally and freely. The various proteins found in the phospholipid bilayer that forms the mosaic pattern are always changing. Because of these two reasons, we can term the plasma membrane as a fluid mosaic model. Question 6. What are the factors that determine the permeability of a substance? Answer. We have two factors, and the answer is size and polarity of the molecules. Question 7. Why is the phospholipid bilayer of the plasma membrane arranged opposing each other? The answer. The characteristics of the phospholipid were the head being hydrophilic and the tail being hydrophobic made it to be arranged opposing each other. Next question. The tail of the phospholipid is hydrophobic and it prevents polar molecules from passing through. Why is water, being a polar molecule, able to pass through the membrane easily? Answer. Water could pass through the plasma membrane easily because water molecule is very small. Size is only factor that affects the water molecule from gaining access. So students, we have already reached the end of this episode. So let's do a recap on what we learned today. Now, in the beginning, we saw the plasma membrane and its structures. We saw how it had a phospholipid bilayer and how there were proteins and other molecules attached to them. We also saw the functions of these proteins, which were for transport and for identity. Very good. And next, we also saw about the phospholipid membrane itself. Now, the bilayer can allow certain molecules to enter directly, which were, for example, water, yes, small lipid molecules. The carrier proteins function as a way of transporting larger molecules such as glucose and amino acids, and the pore proteins acted like a channel that could allow ions or small polar molecules to enter. Now, after seeing those things, we moved on to the phospholipid molecule itself, and we saw that its head and tails had different characteristics: hydrophobic, hydrophilic, and it was because of these characteristics we have a bilayer. And we also saw the selective permeability of a plasma membrane, which allows certain substances to move in and does not allow certain substances to move in. Well, as a conclusion of all that, we also went through a self-assessment, and I guess most of you would have guessed the answers correctly. But if you did not, don't worry, because you can still keep going on. As you repeat it a few times, you'll be able to completely memorize and understand the structures of the plasma membrane. And you got to get it done fast, because next we will be going into something more tougher, something which involves a great level of interest between you. So that's all for today's episode, and thank you for watching iTTV. We'll meet again in our next episode.